Welcome to chapter 8.1. I'm here to tell you about an organism's metabolism. Of an organism's chemical reactions are called metabolism. Number 2. Metabolism is an emergent property of life that arises from interactions between molecules within the orderly environment of the cell. 3. The chemistry of life is organized into metabolic pathways. And that's just so romantic. Metabolic pathways begin with a specific molecule, one molecule, and then you won't guess what's next. It's then altered in a series of defined steps to form a specific product like cereal. So the honey in the bunches are the molecules, and the cereal is a specific product. Thermal energy or heat energy is kinetic and associated with random movement of atoms or molecules. And then this potential energy is the energy that matter, blah, 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 blah. Chemical energy is a form of potential energy stored in molecules because of the arrangement of the atoms. It's kind of like this. These are atoms. This next part's outrageous. A specific enzyme catalyzes each step of the pathway, and then next, catabolic pathways release energy by breaking down complex molecules to simpler compounds. A major pathway of catabolism is cellular respiration, in which the sugar glucose is broken down in the presence of oxygen, carbon dioxide, and water. Anabolic pathways consume energy to build complicated molecules from simpler compounds. These are called biosynthetic pathways, released by catabolic pathways and can be stored and then used to drive these pathways. Now you have to imagine the world
has ended. The major roads are closed, but you can still travel on anabolic pathways.
Energy is fundamental to all metabolism, and therefore the understanding of energy is key to understanding how the living cell works. And we know this because bioenergetics, whatever that is, no one knows what the hell that is really, is the study of how organisms manage their energy resources, hence energetics. I'm energetic to be here on the show. Organisms transform energy, that's obvious. Energy is a capacity to do work, and then it exists in various forms in cell photosynthesis. And it's this green plant, and it's like, oh, we're green, we have chlorophyll, wow. And then the sun is like, oh man, if you look at me, you'll die and you'll go blind, but I'm gonna give you all this energy for plants. That's great. Wally,
What?
I need your help. Can you read this? Can you read this?
Energy can be converted from one form to another.
What? Okay, that was great, Wally. Wally is my son. The energy transformation of life are subject to two laws of thermodynamics. Number 1 states energy can be transferred and transformed, but cannot be created or destroyed. The first law is also known as the principle of conservation of energy. The second law of thermodynamics states that every energy transfer or transformation increases the entropy or chaos of the universe. You can think about this like when your wife cheats on you and she increases the entropy and then she ends up dead. That's the second law of thermodynamics. If you need any more explanation, you're an idiot. So that's all we have for this segment. I hope you learned a lot. Wahoo!
Welcome to Chapter 9, Cellular Respiration. One type of catabolic process, fermentation, leads to the partial degradation of sugars in the absence of oxygen. A more efficient and widespread catabolic process, cellular respiration, of oxygen as a reactant to complete the breakdown of a variety of organic molecules. In eukaryotic cells, mitochondria are the site of most of the processes of cellular respiration, making it the powerhouse of the cell.
Alright, when I give you your birthday, I want to see what the people are on.
One more. Yeah, good job.
Cellular respiration is similar in broad principle to the combustion of gasoline in an automobile engine after oxygen is mixed with hydrocarbon fuel. Food is the fuel for respiration.
What's up, guys? It's Jack here with another one of Jack's food reviews. Me and my friend Hershel are trying gummy bears. Did you know that gummy bears are, in fact, food, which is the fuel for respiration? Enjoy.
The exhaust is carbon dioxide and water. The overall process is organic compounds plus O2 equals CO2 plus H2O plus energy, which is ATP and heat. Carbohydrates, fats, and proteins can all be used as the fuel, but it's most useful to consider glucose.
Hey Andrew, what are you doing? Hey Hershel, I'm tracking my macronutrients. Fat, carbohydrates, and protein.
Dude, what are you doing? Glucose is the only thing important for cellular respiration. Just glucose.
Redox reactions release energy when electrons move closer to electronegative atoms. Catabolic pathways transfer the electrons stored in food molecules, releasing energy that is used to synthesize ATP. Reactions that result in the transfer of one or more electrons from one reactant to another are oxidation-reduction reactions or redox reactions. The loss of electrons is called oxidation. The addition of electrons is called reduction. An easy way to remember this is to remember oil rig. Respiration occurs in three metabolic stages: glycolysis, the citric acid cycle, and the electron transport chain and oxidative phosphorylation. Glycolysis occurs in the cytoplasm. It begins catabolism by breaking glucose into two molecules of pyruvate. The citric acid cycle occurs in the mitochondrial matrix. It completes the breakdown of glucose by oxidizing a derivative of pyruvate to carbon dioxide. During lactic acid fermentation, pyruvate is reduced directly by NADH to form lactate, the ionized form of lactic acid, without release of CO2. Lactic acid fermentation by some fungi and bacteria is used to make cheese and yogurt. Human muscle cells switch from aerobic respiration to lactic acid fermentation to generate ATP when O2 is scarce. The waste product, lactate, may cause muscle fatigue, but ultimately it is converted back to pyruvate in the liver. Under aerobic respiration, the molecule of glycolysis yields 38 ATP, but the same molecule of glucose yields only 2 ATP under anaerobic respiration.
Set. Go. Breaking news here in Apex, North Carolina. We have found a tree performing photosynthesis.
The chloroplasts of plants use a process called photosynthesis to capture light energy from the sun and convert it to chemical energy stored in sugars and other organic molecules.
Here is Davis and Hershel in the field with more.
Thanks, Andrew. We're here in the field with this tree. Autotrophs produce their organic materials from CO2 and other inorganic raw materials that they find from the environment. Autotrophs are the ultimate source of organic compounds for all heterotrophic organisms. Autotrophs are the producers of the biosphere.
Thank you, Hershel. Autotrophs can be separated by the source of energy that drives their metabolism. Photoautotrophs use light as a source of energy to synthesize organic compounds. Photosynthesis occurs in plants, algae, some other produce, and prokaryotes.
This tree!
Chemoautotrophs harvest energy from oxidizing inorganic substances such as sulfur and ammonia.
Not this tree.
Chemoautotrophy is unique to prokaryotes.
Not this tree.
Thanks, Daves and Hershel. Heterotrophs live on organic compounds produced by other organisms. These organisms are the consumers of the biosphere. The most obvious type of heterotrophs feed on other organisms. Animals feed this way.
Other heterotrophs decompose and feed on dead organisms or on organic litter, like feces and fallen leaves. Most fungi and prokaryotes get their nourishment this way. Although no animals are going to eat this tree that they're talking about, they will eat something much smaller, like the grass, down here. Some of our anchors have attempted to depict what is happening in this process. What are you doing, bro?
I'm absorbing light in my chloroplasts, my dude.
Wow.
During this stage, light energy is converted to ATP, chemical energy, and NADPH.
So there are several roles of photolysis. It utilizes the light to split water into the following.
Electrons, which are donated to PS2 to replace the lost electrons.
Hydrogen ions carry the ATP synthase to provide energy for the production of ATP.
And oxygen, which is released into the atmosphere as a byproduct.
Speaking of products, photosynthesis will produce ATP, chemical energy, and NADPH, reducing power and electron donor. Alright, welcome to Chapter 11. So if you were to get into a fight and you get punched, how your body reacts to it is through cell communication. So cell communication is necessary to regulate the cells in the body. It goes from the signal to the transduction pathway, which converts the signal into response through these steps: signal, reception, transduction, response, with ligands, which is the signal, binding for reception. There are 3 different types of chemical signaling. Autocrine is when the cell sends a signal and that same cell responds, like talking to yourself.
Man, I need to use the restroom.
Paracrine is when cells send a signal and the nerve cell responds like shouting, fire.
Paper, scissors, shoot. Rock, paper, scissors, shoot. Rock, paper, scissors, shoot. Hey guys, rock, paper, scissors, shoot. No, you're ugly. Rock, paper, scissors, shoot. Rock, paper, scissors, shoot.
Is when a cell sends a signal a far distance away, which goes through the bloodstream.
Hey. How's it going? Doing great. How about you? Also doing great. Fantastic.
But once in a while, a cell is no longer needed for cell communication and it must be terminated. This is called apoptosis. If the cell is no longer needed, it receives a signal and it dies. It's very safe to do this. Caspase is when the enzymes that make a cell dismantle its DNA and make blebs, which shoot out and slowly break into many pieces.
Hey, how's it going, Mr. Cell? Hi, who are you? I'm Mr. Apoptosis. And you should die. Should I cause collateral damage to the other cells around me, though? Nope. Mr. Caspase? What?
So that is Chapter 11, and on to Chapter 12.
Welcome to Chapter 12.
Welcome. I want you to pay close attention. For what I'm about to do is distort your idea of reality. I will perform mitosis. You follow me, camera guy? We have here Jack. He will be replicated. When I say the magic words, Ms. Johnson, please give me an A. Ready? Ms. Johnson, give me an A.
Yo! You're me, and I'm you! This is so cool.
Unlike what you've seen, this is no magic trick. It's basic anatomy. This process is called mitosis. Mitosis splits a cell into 2 equal copies of itself, creating somatic cells. Somatic cells are all cells that are not directly involved in reproduction. Unicellular organisms use mitosis to divide for reproduction, while multicellular organisms divide for development, growth, and repair. The order of mitosis goes by the acronym IPMAT. Interphase, prophase, metaphase, anaphase, telophase. Interphase is the majority of a cell's life with 3 stages. G1 is for growth, S is synthesis, which copies the DNA, where the cell commits to the idea of reproduction, and G2 finishes up preparing, making sure you're big enough and have enough organelles in the cell. In prophase, the chromosomes bundle into chromatids, and the nuclear membrane breaks down. In metaphase, the chromatids line up in the middle and attach to spindle fibers. In anaphase, the chromosomes are split in half, so the cell is now symmetrical. In telophase, the cell is split down the middle, and the cell creates 2 identical cells. And that's all you need to know about mitosis. Have a great day.
Hey, yo, bro, what it do? Chapter 13, meiosis. Alright, so first thing we gotta talk about is karyotypes. So it's a picture of the chromosomes taken at metaphase. So humans have 23 of them, 23 chromosome pairs, that is. Autosomes are the first 22 pairs, and the sex chromosome is the last pair, because it can either be XX or XY. If you're XY, then you are a male. If you're double X, you're a female. So asexual reproduction are just clones of the parents, and it's very fast. The only variation occurs in mutations. Sexual reproduction is a mix between both the parents, so it's very unique, and there's a lot of genetic variation. Meiosis has 2 cell divisions, creates 2 daughter nuclei, and then they make 2. So you start diploid with 46, and then you finish haploid with 23. Meiosis 1 is crossing over, so you line up double file with their homologous XX. So when they separate, instead of separating chromatids, it separates the homologous chromosome. Lines up the chromosomes up on the metaphase plate, double file with their homolog. Independent assortment, you grab the mom set or the dad set during metaphase, it's randomized. During meiosis 2, separates the chromatids. A mitosis metaphase, same as mitosis, but starts with half the chromatids. And crossing over creates an almost unlimited amount of variation by mixing the mother and father chromosomes together. And you do not copy DNA during meiosis 2 to ensure that we stay haploid.
Yo, what's up? I'm Gregor Mendel, and I study heredity using green peas. This right here is a Punnett square. These 2 letters, those are genotypes. The actual characteristic that's expressed, that's a phenotype. These capital letters, that's a dominant. There's little lowercase letters, that's recessive. Yo, Gregor Mendel back at it. I also created the law of segregation, which states that during gamete formation, alleles segregate from each other, so that each gamete carries only 1 allele. I also created the law of independent assortment, which states that different features segregate independently. This means your hair color, skin color, and eye color don't affect each other. Oh, and by the way,
Get out of here. Yo, I'm not Mendel, and I just want to let you know that not everything follows the Mendelian rules.
oh man, you
Yo,
hit
look
me,
at
bro.
that. Look at that. He's bleeding. That's blood type. That's multiple alleles. That's not Mendelian. Check it out. Yo, guys, over there, I see some codominants. Let's go. See this leaf? It's purple and white. That's codominance, because it shows both the purple and the white characteristic. Wow. Yo, what's up? Yo. Not Mendel. Me and my wife, she's brown. We had a baby, and my baby's tan because I'm pale. This is polygenic inheritance, not Mendel. Yo, welcome to Not Mendel's Cooking. Today I'm gonna whip up some incomplete dominance. See that? Right now it's red and white, but if you mix it, oh, this is kind of gross. One moment. Let's take a cut until I'm done mixing. Yo, and we are back. You know, when you mix ketchup and mayonnaise together, you get a blend of the 2 colors into one color, kind of like a pinkish kind of orange. That's an example of incomplete dominance. And you know, Mendel, I just wanted to say,
Yo, I'm Thomas Hunt Morgan, and I discovered that not all traits follow the law of independent assortment. By studying flies, I learned that gender and eye color are linked traits. A trait that is on a sex chromosome is called a sex-linked trait. 2 traits that are on the same chromosome are known as linked traits. Yo, so I'm chapter 15 right back at you. And parental types are offspring that have the same phenotype as the parents, while recombinant types are offspring with different phenotypes than the parents. For example, in the non-Mendel family that we just saw, Jack had a recombinant type. There he is. That's a recombinant type. And if they had a white baby, like Andrew, that would be a parental type. Here Andrew comes.
Me and my son.
There we are. Happy family. Yo, I'm chapter 15 back at it again, explaining chromosomal alterations. So here we have deletion. See this part right here, the D? Over here, it's gone, deleted. Next we have duplication. Look at this, BC, you see that BC? BC, it's doubled. Now we have inversion. Okay, look, they have that BCD. Now over here, what happened to it? DCB, it's been reversed. That's inversion. Next we have translocation. Look, we have that AB, then we have the MNO, and then what happens is they're flipped. The MNO goes over there, the AB goes over there. The places are reversed, or they take each other's places. You know, that's called translocation. You know, that's our DNA alterations right there. Whoa, bro, I just woke up. I'm chapter 16, how's it going? Replication begins at the origin of replication, where the DNA strands began separating. Separating the two DNA strands creates a replication fork. Replication forks are a Y-shaped region where new DNA strands are elongated. Helicases are enzymes that untwist the double helix. DNA polymerase is responsible for a lot of things in the DNA replication process. DNA polymerase catalyzes the elongation of new DNA, synthesizes the leading strand, and must work in the direction away from the replication fork to elongate the lagging strand. And that's DNA replication. So if you ever need to replicate some DNA, that's all you need to know. So, see you later.
Gene expression is the process by which DNA directs protein synthesis. The first step of gene expression is transcription. Transcription is copying a gene's DNA sequence to make an RNA molecule. This is a strand of DNA, and Hershel is RNA polymerase. There are 3 steps to transcription: initiation, elongation, and termination. In initiation, RNA polymerase, the main enzyme involved in transcription, binds to the promoter with the help of transcription factors. It then begins to separate the strands.
In the second step of transcription, elongation, the RNA polymerase moves along the DNA strand. It builds an RNA molecule by stringing together nucleotides that are complementary to the template strand of the parent DNA. Uracil replaces thymine that is present in DNA. In the third and final step, termination, DNA sequences called terminators signal that transcription is complete.
I am the Terminator.
The new RNA is released from the RNA polymerase.
Hi there, everybody. I'm Bob Ross. Welcome to my studio. Today, we talk about a little something I like to call gene regulation. Gene regulation is the process of controlling which genes in a cell's DNA are expressed. Different cells may express different genes, even though they possess the same DNA. For example, liver cells may produce proteins that neutralize those bad toxins, while neural cells may send signals in our brains via neurotransmitters. Gene expression is affected by internal signals, proteins from the mother cell, damaged DNA for example, and also external signals such as chemical signals from other cells and nutrient levels. Some genes need to be turned on in one part of the body and not in another. All stages of gene expression in us humans can be regulated. Transcription is the main stage of regulation. There are 2 types of gene regulation: transcriptional and post-transcriptional. I'll start with transcriptional gene regulation. Let me just draw you what I mean. transcription factors are proteins that regulate the transcription of genes. They help turn specific genes on or off by binding to the nearby DNA. They are required for RNA polymerase to bind to the DNA and perform the job, transcription. Activators are transcription factors that activate transcription by helping RNA polymerase bind to the promoter of a gene. They're friendly people. But repressors are transcription factors that repress transcription. By blocking RNA polymerase from doing its job. Repressors are bad people. So that's all we need to know for transcriptional gene regulation. Now, post-transcriptional gene regulation, my favorite. RNA processing is the modification of pre-mRNA, such as splicing, capping, and the addition of a poly-A tail, to form mature mRNA that can be used during translation. Alternative slicing is the main post-transcriptional method of gene regulation, and I'm going to demonstrate it for you here. Alternative splicing is the selection of different portions of the mRNA to be used as exons, which are part of the final mRNA, and introns, the pieces to be discarded. I've illustrated for you here alternate splicing. As you can see, in this spliced mRNA, there's a little bit of this lovely crimson color. Look at it. It's such a great color. But in this piece of sliced mRNA, more of that citrus yellow is present in the middle here. Just an example of how exons and introns can be altered in spliced mRNA. There are a few other post-transcriptional gene regulation methods. I'll go over those now. Something called small regulatory RNAs, tiny little microRNAs, whose direct role is to reduce the expression of target genes. And finally, the phosphorylation of proteins, my favorite. The proteins produced by translation can also be phosphorylated. Phosphorylation, which is the addition of phosphate groups, can activate, deactivate, or change the function of a protein. And that's all we have for you today. Remember, beauty is everywhere. A virus is an infectious particle that reproduces by commandeering a host cell and using its machinery to make more viruses. Because they cannot reproduce on their own, viruses are not considered living. They utilize the same genetic code that is present in the cells of all living creatures. They attach to a host, insert their genetic material, genome is expressed, then new viruses are assembled and they exit the host. A bacteriophage is a virus which infects a bacteria and uses it to produce more viruses. It infects bacterial hosts using one of two cycles. In the lytic cycle, the phage infects the bacterium, hijacks it to produce more phages. Now, the newly created viruses are inside the bacterial cell. The virus kills the bacterium by causing it to explode, or lyse, releasing the newly created viruses. At the end of the lytic cycle, the bacterial cell was sadly destroyed, so we're back with a whole new bacterial cell. We're going to show you the second type of viral infection of a bacterial host cell, which is called the lysogenic cycle. In the lysogenic cycle, the phage infects the bacterium once again. It inserts its DNA into the bacterial chromosome. And this allows the bacterial chromosome's newly inserted viral DNA to be copied and passed down by the bacterium, so that this cell and all the cell's offspring can continue to produce viral pieces. Isn't that sad?
Wait, what's this magic trick stuff? What am I supposed to say about that?
No, with the... Oh, God.
Alright, get out. Get out now.
Mayonnaise.
No, get out. We're done.
What?
Get out of the frame.
You're actually going to cut.
Get out of the frame, boy.
No, you're just gonna cut the.
No, I'm just gonna cut
mayonnaise.
I don't need more mayonnaise. No, that's more mixing for me to do. Stop it. You see, once you mix the red and white, it goes orange. It's incomplete dominance.
Hey, Drew.
What'd I do?
What does red and white make?
Did I say orange?
Yes!
Yeah,
Did it not work?
conjoined twin. Alright, I'll stand like over here. Okay.
Right here.
Alright, keep it like that. Alright, hold up.
I like it. I like it.
So please.
That's
really sick reaction.
That's great.
If that actually happened to me, I'd like freak out.
Hello, Wrigley.
Wait, why did you try it? Hershel, you finish.
Hershel. Somebody once told me the world is gonna roll me. I ain't the sharpest tool in the shed. She was looking kinda dumb with her finger and her thumb in the shape of an L on her forehead. Well, the years start coming, they don't stop coming. Fed to the rules and I hit the ground running. Didn't make sense not to live for fun. Your brain gets smart, but your head gets dumb. So much you do, so much you see. So what's wrong with taking the back streets? You never know if you don't go. You never shine if you don't glow. Hey now, you're an all-star. Get your game on, go play. Hey now, you're a rock star. Get the show on, get paid. And all that glitters is gold. Only shooting stars break the mold. It's a cool place, and they say it gets colder. We bundle up now, wait till you get older. But the media men beg to differ, judging by the hole in the satellite picture. The ice we skate is getting pretty thin. The water's getting warm, so you might as well swim. My world's on fire, how about yours? That's the way I like it, I'll never get bored. Hey now, you're an all-star. Get your game on, go play. Hey now, you're a rock star.